I'm going to talk a little bit now about platelets. So platelets, they're also called thrombocytes and are fragments of much larger cells called megakaryocytes. Megakaryocytes develop in the bone marrow and they fragment to form platelets, which they don't have their own nuclei, so they can't reproduce, but they have many other functional characteristics that are vital for haemostasis, and I want to go through them now. Now platelets have three important roles which I like to describe as the three A's. The first is adhesion. Now, as I mentioned in the overview of coagulation, adhesion is when platelets come into contact with the subendothelial tissues, particularly collagen, and stick to it. And this is an important process in initiating much of the rest of coagulation, and is the first step in platelet function. The second is activation, both through the process of adhesion and through other factors which include positive feedback from other activated platelets themselves, and from, for instance, thrombin, which is produced as a product of the coagulation cascade. Platelets become activated, and when they become activated, this allows release of procoagulant factors from the platelets, which continues the positive feedback mechanisms that have already been started to promote further platelet action and promote the coagulation cascade. And in a sense, they also allow the third part of platelet function, which is aggregation. Aggregation is the final step in platelet action, which is the beginning of platelets to start to stick to each other. They start by sticking to the platelet wall, but they start by sticking to the subendothelial tissues. They become activated, and become activated both from adhesion and from other factors that I mentioned. And then finally, once activated, they are able to start to stick to each other in a process called aggregation. And once aggregation is ongoing, they're forming the primary platelet plug, which we've discussed before is when blood loss starts to be stopped. So the first characteristic of platelets that I want to describe is one of the most important, and it's the concept of glycoproteins. So glycoproteins, they exist like this on the surface membrane of platelets, and are very important in all three stages, all three roles of platelet function. So I'm going to spend a bit of time describing them now. So glycoproteins, and there's three different types of glycoproteins which are important. First, glycoprotein 1A. Second, glycoprotein 1B, and the third and most important, glycoprotein 2B 3A. These are all on the surface membrane of the platelet, but there's a third glycoprotein which is actually produced in the endothelial cells, and it's also present inside platelets, but it plays a role as a cofactor for allowing these glycoproteins to function, and it's called von Willebrand factor. Von Willebrand factor. And this is a large glycoprotein which it exists in multimers of different sizes, and as I said, it's produced mainly from the endothelial cells and is released constantly into the plasma. But it also can be released by platelets when they become activated, and it has a role as a cofactor for allowing these glycoproteins to function as in the way of the three A's, as I'll show you now. So what we'll do is we'll give a visual representation of how they work. Here's our platelet membrane there, Here's our collagen fibres that have been exposed by the vessel wall injury. And to start with, we've got a glycoprotein. So this is glycoprotein 1A. And glycoprotein 1A is very straightforward. It can adhere directly to the collagen. It allows adhesion to the collagen fibres without any further processes to go on. Secondly, we've got glycoprotein 1B. Now, this glycoprotein requires the presence of that cofactor I mentioned, von Willebrand factor, and it needs this protein to be present to allow any sort of adhesion to the collagen fibres. And this protein is an important part of adhesion as such, and there is indeed a condition where von Willebrand factor is not present, and it affects platelets' ability to adhere to the collagen fibres. So I've discussed the first two glycoproteins and their obvious actions in allowing adhesion to the subendothelial tissues. However, these both also allow activation of the platelets through this adhesion process. By contact with the collagen fibres, the platelets become activated primarily through the role of calcium, and this activation process is also promoted by the other factors I mentioned before: thromboxane A2, ADP, thrombin, and it allows the platelets to undergo various changes. It includes secretion of other procoagulant factors, but with relation to what I'm discussing now, it allows upregulation of the third and most important type of glycoprotein, glycoprotein 2A, sorry, 2B, 3A. And I'll tell you a bit more about this glycoprotein. It's usually present on the surface of inactive platelets, but when the platelet's inactive, this glycoprotein is pretty inactive itself as well. When platelets become activated, as for instance via adhesion to collagen, these glycoproteins undergo upregulation and they increase in number. One of their roles is they are able to adhere to von Willebrand factor in the same way that glycoprotein 1B can, and increase adhesion. But their most important factor is related to how they interact with fibrin. Once the plate's been activated, these glycoproteins are the ones that attach to the fibrin filaments, and through the intermediary of these fibrin filaments, these glycoproteins are therefore able to connect with other GP 2B 3A receptors on other platelets. And as we can see here, this is when platelets are starting to stick to each other, and therefore the process of aggregation is occurring, which hasn't occurred through these other glycoproteins. So through the action of this glycoprotein, platelets can move from attaching themselves to the subendothelial tissues to actually attaching themselves to each other, and this is a very important step in forming the platelet plug. So I'm going to summarise this now. We've got our glycoproteins here, we've got the different types, and the first two have roles in adhesion to the subendothelial tissues and also in actually activating the platelets. Now, those are two very important points in platelet action, but the third step involves glycoprotein 2B 3A, which is only possible once platelets have been activated. These are able, through attachment to fibrin filaments allow aggregation of platelets, attaching to other platelets through the same glycoprotein, and thus through these glycoproteins the platelet plug can be formed. This is a very important glycoprotein, and as such is the target of certain anticoagulant medications, because without this, yes, you've got adhesion and activation of the platelets through the first two glycoproteins, but you need platelets to aggregate together to form the platelet plug and to start the whole process of actually forming a final fibrin clot.